Okay. We are now going to take this the next level. There are things that Gregor Mendel simply could not have told us. He told us plenty with what he had, but he was working with a really simple system, and that's a good thing because likely he would not have been able to discern the laws that he did if he had been working with something more complex. But of course, we know that not all genes are a simple on and off, as we see so many of them in pea plants. The relationship between your genes and what your phenotype is usually is much, much more complex than what we see in many of these pea plant traits that Mendel was looking at. So what we're going to do is go that step beyond what Mendel did, actually several steps beyond, and understanding now how so many traits they are so much more complex than what he originally saw in his pea plants. Okay, so first thing to remember is that what Mendel showed us with his pea plants and his monohybrid crosses and dihybrid crosses and all that was he showed us something called complete dominance, in which one trait would completely dominate another trait, a recessive trait. So you would have this particular hereditary pattern that would result because of that. Well, there are, in fact, other types of dominance. Indeed, there is, since we have complete, of course, you know, we're going to have incomplete dominance, which does show us, interestingly, that blended phenotype that was so popular in Mendel's day. It does exist. It just does not exist as the explanation for every type of inheritance. But there are times when you will blend one type, perhaps a red flower with a white, and you will get an intermediate phenotype that looks blended. We would think of that perhaps as our blending our dominant red with our recessive white and instead getting pink. But understand that's not what it is, because these are equals now. One is not dominant over the other. So red and white does not subordinate to red. It's equal. So we need a different notation. Okay? So now with incomplete dominance, we would still call the red flower RR because it's red, but we would show the white flower as being labeled WW because it is not recessive anymore. We're not going to use those lowercase letters, which means the pink flower would now have the genotype RW. Okay? And that's really pretty simple. And what's really interesting about incomplete dominance is now we can see those heterozygotes. We can see them, and they show up the same. The ratios between the genotypic ratio and the phenotypic ratio are now always going to be the same in this type of dominance because the heterozygote does in fact look different. If you have a true breeding red and a true breeding white and they show incomplete dominance, then what we're going to see is all pink flowers in that F1 because they will all be RW. And then in the F2, when these self-pollinate, we will see a 1 to 2 to 1 phenotypic ratio because now we are seeing that heterozygote. We don't have that 3 to 1 phenotypic ratio anymore because the heterozygote actually looks different. Okay? Codominance is another type of dominance, and this time the alleles involved are equals like in incomplete dominance, but they are not blended. They are expressed separately in the organism. You need to think perhaps instead of a pink flower, a red and a white giving a red and white striped flower or something like that. The classic example is human blood typing, the ABO blood groups. As you may be aware, humans have you can have type A blood, you can have type B blood, you can have type O blood, and you can have type AB. And all this is referring to is the proteins, and this is kind of trying to show you an example of that, the proteins on the surfaces of your blood cells. There's one protein or antigen, that's another thing for markers on the cell surfaces of things. Antigens on the surface of a blood cell that look one particular way, like this, we might say are type A antigens, and so that's what your markers look like. If you're a type B person, oh, I don't know, maybe they look like this. They look different somehow, okay? If you are a type O, you simply lack antigens. You have nothing. But if you're type AB, you have both. And that is the codominant situation. Whoops, sorry, I wrote a B instead of drawing my little square. Okay, that is where these show up separately on the surface of the cell. They look just like they look when they're alone. They show up separately. They are equal. Okay, that is codominance. So what this should maybe suggest is that in the human population, there are actually 3 different alleles for blood type. This is an example of multiple alleles, where there are multiple alleles that exist in the population for a given trait. Now, how many of those alleles are you allowed to have as an individual? You only get to have 2. Why? Because you're going to get one from your mother and one from your father. And unless you have some weird third parent, you're not going to get 3. You're only going to get 2. So, how this works, the symbols we use for the genes, for the alleles for human blood are IA, IB, and little I. I simply is for the word immunoglobulin, which is what the general name for these little proteins is. So if you are a type A person, okay, you have at least 1 copy of this type A allele for blood type. And if you've got 2 copies, 1 from each parent, then you're type A, you're homozygous. If you got a type A allele from one parent and a little I from another, this little I is little to indicate that it's recessive. Then you're still type A. IA is the A allele, IB is the B allele, little I is the O allele. So, if you're a type B person, then you could get IB from both parents and be homozygous, or you could get B from one parent and an O allele, little I, from another, and because this dominates that, you'd still be type B. To be a type O person, you could only be that way by getting a little I from each parent because it's recessive. If anything else is there, you're going to have the other blood type. And AB simply means you got an A allele from one parent and a B allele from the other, and that's the only way you can be AB. Okay, so A and B are codominant to each other, and they are both dominant over the little I. Little I is recessive to both of them. So, this is just kind of going over what we just did. Either of these genotypes will give you that phenotype. Either of these genotypes will give you that phenotype. This is the only genotype that will give you that phenotype. And interestingly, this person, because they have both of these, then their immune system is programmed to understand both of those. They can receive any blood in a case of emergency. They are a universal recipient. Type O blood, unfortunately, is only the universal donor. They recognize no antigens because they don't have any. So if they were to get either of these blood types, their body would mount an immune response, and it would be very bad. They can only receive blood from another type O person, but they can give to anyone because they possess no antigens to trigger an immune response. And of course, these people can only receive or give type A. These people can only receive or give I'm sorry, they can only receive type A or type O. They can receive type B or type O. Okay. Pleiotropy is the term for a gene that affects more than one physical trait. Dwarfism is a great example. It's one gene that causes dwarfism, but it affects many, many characteristics of someone who's a dwarf. There's many characteristics that cause that condition. Obviously, the short stature, but there's also issues about muscle tone and some other issues that is caused by this one gene being wrong. So that's pleiotropy. Okay? Same thing goes for gigantism. It's one gene that causes it, but it has many, many, many effects on the individual. Okay? Interestingly, achondroplasia, dwarfism, is inherited as a dominant trait. We often think of abnormal genes as being recessive, but they are not always, and that's important to remember, and they cause different inheritance patterns. In a dwarf situation, if you have the big A, then you have the disorder. If you are little a, then you're normal. So all of us that are of normal stature are little a genetically. It is this dominant gene that causes the disorder. Okay? So, is it possible? Well, let's look at this side first. If we cross a dwarf with a normal person, what would the results be? Can they have normal children? Can they have dwarf children? The answer is yes on both counts. In fact, 50% of their children would be dwarf and 50% would be normal. The normal parent would always donate this. 50% of the time, however, the dwarf parent would donate that. So 50% of the time you'd have a dwarf offspring. Can two dwarves have a normal child? Yes, they can. How? Because a dwarf is a heterozygote by definition. So they can end up with a normal child because each of them could donate a little A. Interestingly, the homozygous dominant condition is actually lethal. You cannot survive with that condition for more than a very, very short period of time. Most babies that are like that die shortly after birth. Okay? That means if you are asked a question, how can I phrase? Two dwarves are going to have 3 children. What are the odds of one of them being normal? You cannot count this child in that equation because it would not survive. So, in other words, the odds of them having a normal child in reality are 1/3 because we don't count this child as being part of the equation. Epistasis is another, or epistasis, I think is the proper pronunciation, is when one gene completely blocks the expression of another gene. Class example is coat color in mice. It is controlled by 2 separate genes. One gene simply controls for will there be pigment or will there be no pigment placed in these cells. The other will tell you how much. Big B means a lot and the mouse would be black. Little b means not as much and the mouse would be brown. Okay? So, if you are little c, I don't care what your B combination is, no pigment is going to be placed. So you're going to be albino. So this combination right here would give you a black mouse. This combination would give you a brown mouse. Okay? But this combination, I don't care what's in front of it, is always going to give you an albino mouse, okay? Because we won't allow any pigment to be laid down. And that gives you a really weird dihybrid combination. It gives you this weird 9:3:4 ratio, as opposed to a 9:3:3:1 that we typically are used to seeing. Same thing in Labrador retrievers. If you like dogs, then here's an interesting little tidbit. Two genes involved in color. Again, we have this pigment or no pigment gene, and we have the pigment amount gene in the fur. So what we see is in this dog, for example, we are going to allow pigment to be laid down, and boy, that pigment's going to be dark. So this animal is going to be a traditional black lab, black fur, black nose, the whole bit. This animal is going to have a lighter concentration, but we are going to allow the pigment to be laid down, so we get the brown fur and the lighter nose and all of that. In this case, we are going to see no pigment being allowed in the fur, but we will see the black nose and lips and eye rims and stuff, because this only affects, the E only affects the fur, not the skin. Over here, we will see the no pigment in the fur again, but we will see the brown nose, eyes, and all of that. So, another way that genes can have different patterns than what Mendel showed us is you can have many genes affecting one trait, and that is called polygenic many genes. Classic example is human skin color, but also other traits as well. Here we have at least 3 gene pairs contributing to skin color, and what that results in is most people having something in the middle, but you can have extremes on either end, and what you end up with is this continuum of all kinds of skin colors. There's no light, dark, period. There's all kinds of combinations in between. And the same goes for height and weight, all kinds of human traits and traits in other organisms as well, where many traits are feeding into it, give you this continuous variation pattern. And don't forget, the environment can affect all these as well. If you're out in the sun a lot, your skin color is going to be different than someone who is not. If you are not eating well, you're not going to attain the same height that you might if you had a good diet, and so on. So environmental roles can play a part. Albinism, however, is a different kind of thing in that one gene pair can determine if you're going to make pigment or not. Excuse me. Melanin is the pigment that colors everything. It's just whether do you have a lot of it or a little of it in terms of your skin color. Melanin is the pigment. Tyrosine is an enzyme that gets converted to melanin, but if you don't have the enzyme to make that conversion, then you don't get a melanin, and then you have this albinism that we see in these individuals. That's a one gene mistake right there, just not making that enzyme. That's a one gene mistake. All right, sex linkage. There are 2 sorts of chromosomes. You've got autosomal chromosomes and sex chromosomes. This fact was first discovered by Thomas Hunt Morgan at Columbia University, and what allowed him, he actually is the first person to actually link genes to the chromosomes and say this is where the genes or Mendel's factors are found. And what led him to that was the fact that he was working with fruit flies, and he got this weird mutant fruit fly one time that had white eyes, so he started crossing it with a normal fruit fly that has traditional red wild type eyes. And as he watched the hereditary patterns, he said, you know what? This trait is behaving just as though it were inherited on the on the X chromosome, rather, on the X chromosome. And he could figure this out because, unlike Mendel, he did have microscopes to see these chromosomes, and he knew how they behaved at meiosis. So, this is what he noticed. He noticed if he had a true breeding red-eyed female and one of these mutants that was male and it was also true breeding, if he bred them, he would get all red-eyed flies, males and females. The white-eyed trait would seem to go away. Then if he crossed those with each other in the F1, all of the females he got, all of them would be red-eyed. But among the males, he would get this 50-50 split. He would get 50% red and 50% white. Why? Why were they only showing up in the males, this white-eyed trait? Sex must make a difference with this. So, what what Morgan figured out essentially was what we know in a lot more detail now. Of course, we know there are two X chromosomes in humans and in fruit flies, and there's in human females, two X's are what make you female. X and Y, however, are going to make you male. So if you have a male and a female, you have a 50-50 shot, of course, of having females or male children. It's 50-50. Who makes the determination? The male does, because the female is always going to send an X. The male is going to send either an X or a Y. Now, what you need to notice is, of course, the females are homologous. The X chromosomes are homologous. If the gene for eye color is here, the gene for eye color is also going to be over there. But in males, if the gene for eye color is here, there is nothing comparable on the Y. So whatever the gene for eye color is here is what he will have. Whereas if the female has a weird recessive trait on one chromosome, there's always the chance it could be blocked by a dominant trait on this chromosome or masked by a dominant trait on that chromosome. We can't mask anything with the male. If it's on here, and then it's going to be expressed. Okay? So knowing that, let's think about Morgan's fruit flies. So he's got this completely normal red-eyed female, and he's crossing her with this completely pure breeding white-eyed male. Okay, and notice our notation. We have to note the X and Y because they are relevant. So this means the female is homozygous, dominant. This male is homozygous, is recessive, and he only has the one R because you can't have an R on the Y. We cross them and we get this combination. We get a female that's red-eyed, a female that's red-eyed, a male that's red-eyed, a male that's red-eyed. They're all red-eyed. Now, we're going to cross two of these. But this time what we're crossing is not pure breeding. We've got this heterozygous female crossing it with a pure red male. Here's what we get. This child, this offspring, would get the big R from both parents, and it would get an X, so it would be a red-eyed female. This offspring would get the Y from its dad and the R from its mother, the XR from its mother, and it would be a red-eyed male. This offspring would get the recessive from mom and the dominant from dad, and it would be still a red-eyed female, though it would carry the gene. And here is your white-eyed male. And this is why Morgan would only see white-eyed males, because the females stood a much better chance of having getting a gene that would mask that, whereas the male can't mask it. If he's got this, there's nothing comparable on the Y to mask it, and he is always going to show it. Okay? So, That's sex-linked traits. What is on the Y chromosome then? Well, not much other than a gene that determines maleness. This gene, the SRY gene, is the master gene for maleness, and it turns on all the genes that are going to cause male hormones to be produced. Again, a great example of pleiotropy. One gene has many effects. Okay? The X chromosome has tons of genes on it, including tons of genes that have absolutely nothing to do with sex. And mutations in some of these genes can cause really not good conditions. Hemophilia, where your blood will not clot, a type of muscular dystrophy seen mostly in males, color blindness. Interestingly, all of these traits are seen even though they are on the X, and because they are on the X, they are seen the vast majority of the time in males. It's not that a female can never be colorblind. She can, but she has to inherit it from both her father and her mother. She has to look like that. Whereas a male just gets it from the mom, and he always gets his Y from his dad. So here is a human X chromosome. This is showing all the different disorder genes that are associated with it. I mean, these are all normal genes, but something goes wrong with them and they can cause a disorder. More than 60 diseases are linked to the X chromosome. The Y, on the other hand, has basically just one or a few significant genes, the most significant being the SRY. It's the main thing. There are lots of other genes on there that make a male too. Sometime when you have a little time, you can check those out. All right. That's a pedigree for a disease called hemophilia, which is sex-linked. Please note, we can't write hemophilia like this because it is sex-linked. It's on the X, so we have to write it like this. And we always indicate a female with a circle and a male with a square when we're doing a pedigree. And we'll talk more about pedigrees in class, but suffice it to say that this is very much like the eye color in the fruit flies. A female who is heterozygous and a male who's normal run the risk of having, well, here, let's look. Normal female, normal male, normal female. She's going to have the normal gene, but she will be a carrier, and who's going to get it is a poor male. He's the only one that's going to really show the effects of this. Okay? And there we go, highlighting that she can pass it on to her sons later on. Again, she can pass it on to daughters too, but she would have to marry a man who had hemophilia, and he would also have to give it to a daughter in order for her to get it. All right, almost done. X inactivation. Because a female has two X's, two X chromosomes, but really, as we can see in males, all you need is one X in order to survive. That's it, because males survive with one X all the time in this little puny Y. Females don't need both X's, and indeed, one of them gets inactivated. Okay? One gets inactivated during development when a female is an embryo. Here she is as a zygote. She's got her two X's. This is a calico kitty cat, by the way. And let's say the fur color is associated with the X chromosome. So let's say we've got the orange allele and the black allele, and you can see that there. One X has the orange, one X has the black. Now, during embryonic development, one or the other gets inactivated, and it's at random in these different cells. You can see in this one, the orange chromosome got inactivated, but here the black one got inactivated. Now, this inactivated X chromosome is called a bar body, and if you looked at any female in our classroom and her cells, you could see, if you had a good enough microscope, these X's balled up and just tucked away on the side of the nucleus. Okay? It is completely random which one is going to become the bar body. Okay? And how that works is all the cells that this embryonic cell gives rise to, this one will be the one that's inactivated. And all the cells that this cell gives rise to, this one will be the one that's inactivated. And it works out that you end up with this mosaic of patchwork in the tissue. Okay? And that's how come calico cats look the way they do. They get these patches of black, patches of orange, and the white is due to a different kind of genetic thing going on. And that is also why only females can be calicos. If you see a calico cat, you know it's a female because they are the only ones that can get this patchwork type configuration due to the inactivation of one or other of the X at random. Okay? Male pattern baldness. There are things called sex-influenced traits. Okay? This baldness is an autosomal trait, but it's influenced by sex hormones, and that's why we see it more often in males than females. The condition is dominant in males and it is recessive in females, and it is due to interactions with the sex hormones. This combination, either big B or big B, little b, will lead to baldness in males, but you have to have both of these to become bald in a female. Okay? Environmental effects, we already talked about those, that environmental effects can also affect the expression of genes. The type of soil can affect the color of flowers. Obviously, the climate can affect the color of the Arctic fox. So environmental factors are also really important. And that was really long, and that's it. And I hope you broke this up into 2 parts because you're going to kill me if you didn't. Hope you have a great evening, and I'll see you soon. Bye.